Okay, let's continue with chapter 8, cell reproduction. Again, to meiosis. In this video, so it's going to be 106 cell repro 3. I don't know whether we'll finish it. I may have to make a cell repro 4. Well, we'll see. So, meiosis is sexual cell division, and it's quite specialized compared to mitosis. It's only it is only conducted in gonads, in ovaries or testes. That's it. It's the only place it happens. And the cells that conduct it are either called spermatogonia, spermatogonium, singular, or oogonium, or oogonia, plural. And they are diploid cells. Everybody thinks that they're haploid. No, they're diploid. It starts with, so we go, mitosis goes from diploid cells to diploid cells. Meiosis goes from diploid cells to haploid cells. Now, in a spermatogonia or oogonia, the chromosomes duplicate in interphase, in an S phase. So they go through a regular cell cycle. And the chromosomes replicate in the S phase, okay, of interphase, or chromosome duplicate. And what's going to happen is homologous chromosomes are going to find each other in meiosis, and they are going to be segregated, put into different cells, separated into different cells in meiosis 1. See, that's right. Meiosis is a two-stage process. Meiosis 1 is meiosis 2. And meiosis 1 has the same phases as mitosis, but you have to put a Roman numeral after it for the phases of meiosis. So it's prophase 1, so meiosis 1 is prophase 1, metaphase 1, interphase 1, telophase 1. Then it's a short interphase without any more DNA replication. And then you have meiosis 2. So then you have prophase 2, metaphase 2, interphase 2, and telophase 2. So in meiosis 1, homologous chromosomes separate. And this is one of the big differences between mitosis and meiosis. So you need to know the differences between the two processes. One of the big differences is tetrads are formed. A tetrad is a pair of homologous chromosomes. And then a tetrad separate in meiosis 1. And that doesn't happen in mitosis. Okay. Now, so after the end of meiosis 1, you actually already have haploid cells. If it's a human cell, you'd have 23 chromosomes when you started with 46. So why not stop there? Well, here's the thing. There's 23 chromosomes, but they're replicated chromosomes, right? And gametes have 23 single chromosomes. So what we have to do is go through meiosis 2. And meiosis 2 is really exactly the same as mitosis. It's just done with a haploid number of chromosomes. So the replicated chromosomes split apart. The centromeres are holding together, split apart. And the replicated chromosomes become single chromosomes. And so at the end of meiosis 2, you have 4 functional gametes. That's true for males. We're going to see that the process is essentially the same between males and females, but there's a difference in the timing, how often it occurs, and how many functional gametes you get. And so we'll get into this later on when we get into, we learn the basics of meiosis first, and then we'll get into the differences between males and females. Okay, process of cell division that produces haploid gametes in diploid organisms. It resembles mitosis, but with 2 differences. The first difference is that the number of chromosomes during meiosis is cut in half. So in meiosis, a cell that has duplicated its chromosomes undergoes 2 consecutive divisions called meiosis 1 and meiosis 2. Because 1 duplication of chromosomes is followed by 2 divisions, each of the 4 daughter cells resulting in meiosis has a haploid set of chromosomes. Haploid set of single chromosomes. Okay, the second difference with meiosis compared with mitosis is an exchange of genetic material, pieces of chromosomes between homologous chromosomes, we should say non-sister chromatids, exchange pieces of each other. It's called crossing over, or it's also called recombination. So you scramble your chromosomes. Chromosomes are scrambled in this process. And to actually make sure that you don't give your kids the same chromosomes you got from your parents, you're going to scramble your parents' chromosomes before you give them to your kids. It happens every time meiosis happens. Okay. So here's the interphase, nuclear envelope on condensed chromosomes. Chromosomes duplicate in the S phase, right? Then prophase 1. Homologous chromosomes find each other. How do they find each other? I have no idea, but they do. They find each other, and crossover occurs. Now you see on these diagrams, one set of homologous chromosomes is blue. Let's say that's from dad, and the red's from mom. And so you have a blue chromosome and a red chromosome. Well, they cross over, and pieces of the red go to the blue, and pieces of blue go to the red. They cross over the same place and exchange equal pieces of chromosomes and exchange segments called crossing over or recombination. Then homologous chromosomes line up. So that's one big difference in prophase 1 versus prophase of mitosis. In metaphase 1, homologous chromosomes line up in the middle of the cell. It's not the replicated chromosomes that line up, like in mitosis. It's tetrads. It's pairs of homologous chromosomes line up. In anaphase 1, homologous pairs separate. In telophase 1 cytokinesis, two separate cells are made, pinches together if it's an animal cell, or forms a cell plate if it's a plant cell. And remember, this goes on in plant cells too, because remember, plants have genders, they're male and female also. So you end up with two cells with a haploid number of replicated chromosomes after telophase 1. Then we have a short interphase. So here it is again, crossing over in prophase 1. Homologous pairs, tetrads lining up in phase 1, tetrads separating anaphase 1, telophase 1, and cytokinesis. Then, in prophase 2, we have the haploid number of replicated chromosomes, and they're just a jumble, and spindle fibers are forming, and they attach to these chromosomes, and they yank them and pull them into the middle of the cell. Metaphase 2 looks a lot like metaphase of mitosis, because it's a single line of cells with a haploid number. In mitosis, it would be 46 human. In metaphase 2, it's 23. And notice they're mixtures of red and blue, because crossing over occurred in meiosis 1. Then the centromere split in phase 2, and these chromatids separate and go to the poles of the cell. And then in telophase 2 and cytokinesis, you have a pinching together of its animal cells, and you have two cells now, haploid number, single chromosomes. And then meiosis is over. And then you have mature gametes. Those prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis. Okay, for both mitosis and meiosis, the chromosomes duplicate only once in the preceding interphase. The number of cell divisions varies. Mitosis involves one division in the nucleus and cytoplasm, duplication and division in half, producing two diploid cells. Meiosis entails two nuclear and cytoplasmic divisions, duplication, division in half again, yielding 4 haploid cells. All the events unique to meiosis occurring during meiosis 1, really. And as I just mentioned, meiosis 2 is virtually identical to mitosis in that it separates sister chromatids, but it's with the haploid number of chromosomes. Meiosis II yields daughter cells with a haploid set of chromosomes. Okay, haploid set of single chromosomes. So there's the comparison right there of mitosis on the left and meiosis on the right. And so like I said, the unique things in meiosis happens during meiosis I, crossing over and tetrad formation. Okay, offspring of sexual reproduction are genetically different from their parents and one another. How does meiosis produce such genetic variation? Still a little too hot. Okay. Figure 816 illustrates one way in which meiosis contributes to genetic variability. When aligned during metaphase 1 of meiosis, the side-by-side orientation of each homologous pair of chromosomes is a matter of chance. Well, what does that mean? Well, you know, in prophase 1, no, metaphase 1, homologous pair of twins are going to line up. They find each other in prophase 1, they line up in the middle cell in metaphase 1. But, and you know, each pair of homologous chromosomes, we have a maternal pair and a paternal pair, right? Well, what side does the paternal chromosomes go? And what side does the maternal? So you have these two chromosomes. They line up like this or like this. The next pair lines up like this or like this or like this. See what I mean? Which chromosome of a homologous pair goes to the right? Which one goes to the left? Out of the 23 pairs. It's completely random, right? This is called independent assortment. And there are about 8 million different ways 23 pairs of chromosomes could line up. Sounds like a big number, but it's small compared to the other. So there's like a 1 in 8 million chance that you're going to give your offspring the same chromosome sets you got from your if crossing over didn't occur. But crossing over really scrambles things. But anyway, so for a species with more than 2 pairs of chromosomes, such as humans, every chromosome pair orients independently of all the others in metaphase 1. That's why it's called independent assortment. So there's it's just a scenario here with only 4 chromosomes, 2 homologous pairs. They can line up with both blue ones to the left and both red ones to the right. And then you get the combination A and B. Or you have one where the first one, the blue chromosome lines up to the left and the right, red to the right. And in the other set, the right goes on to the left and the blue goes to the right. And so you have different combinations. These combinations, and that would make combination C, combination D. So if you have 4 chromosomes, you have 8 different combinations that could end up. But with 23, it's like, so that's really 2 to the 3rd. It's like 2 to the 23rd power in humans with 23 pairs. So that's about 8 million. Okay. Well, there it is for human. N is equal to 23. So there are 2 to the 23rd power, about 8 million possible chromosome combinations that can appear in gametes. So a single man and a single woman can produce zygotes with 64 trillion combinations of chromosomes. That's a big number. All right. Second thing, crossing over, exchange of corresponding segments between non-sister chromatids of homologous chromosomes occurs during prophase 1 of meiosis. With crossing over, gametes arise with chromosomes that are partly from the mother and partly from the father. So even with independent assortment, there will be a really slim chance you'd be giving your kids the same set of chromosomes you got from your parents. But with crossing over, that's not possible at all because you're scrambling. Each chromosome is going to be a mixture now of mom's chromosome and dad's chromosome. So you really scramble things up. And so, so you make sure that every offspring is genetically unique, genetically different. So, so here's a site of crossing over homologous non-sister chromatids exchange corresponding things. Metaphase 1 sister chromatids remain joined at their centromeres. And we end up with metaphase 2, and you end up with recombinant chromosomes. So these are recombinant chromosomes because when it became recombined, crossing over has occurred. These two in the middle here that are a little bit of red and a little bit of blue. Here's all blue, here's all red. They weren't recombined. The ones in the middle have been recombined. Oh, and which set of non-sister chromatids cross over? Well, that's completely random also. You could have the two inside ones cross over, the two outside ones cross over, the inside one here and the outside one over here, or the inside one here and the outside one over here. Doesn't matter. It's completely random. And how many crossover events you have and what's particular non-sister chromatids are crossing over is like completely random. It's all a crapshoot. It's completely random, but it always happens. Okay, although some species can reproduce asexually, only very few animals reproduce only asexually. And this is Delloid rotifers. They're really tiny animals. They're in a whole phylum all to themselves, rotifera. And they're aquatic. They live in fresh water. They're pronounced Delloid because the B is silent. How would you pronounce BD? Well, the B is silent. Does this entire class of animals reproduce solely by asexual means? There's a rotifer. And as a matter of fact, are we doing that? There's a lab that we used to do with looking at communities, and when we get some pond water and we look at organisms in pond water, and we'd invariably see rotifers. They're quite common, pond animals. So they have thrived for millions of years without sexually reproducing. So Delloid rotifers would display much more variation in their pairs of homologous genes than most organisms, because crossing over reduces actually the variability of homologous genes. It increases the variability of chromosomes, but it makes genes a little bit more homologous. So researchers compare the sequences of a particular gene in Delloid and non-Delloid rotifers. And the prediction would be that there'd be more variation in this one particular gene in Delloid rotifers than in non-Delloid rotifers, because they don't do sexual reproduction. So among non-Delloid rotifers that reproduce sexually, the two non-homologous versions of the gene were nearly identical, differing only by 0.5% on average. In contrast, the two versions of the same gene in Delloid rotifers differed by 3.5 to 5%. These data provide strong evidence that Delloid rotifers have evolved for millions of years without any sexual reproduction. Okay, so what happens when meiosis goes awry? What happens when there's an error in the process of meiosis? It's a mistake can result in genetic abnormalities that range from mild to severe to fatal. So we're going to look at errors in meiosis now. One error is called non-disjunction. Well, first of all, what's disjunction? Disjunction is either the separation of homologous pairs in interphase 1 of meiosis, or the separation of sister chromatids in interphase 2. Either one. That would be disjunction. And that's the normal thing to happen. What happens in non-disjunction is the homologous pairs, they don't separate. Both of them go into one cell. So one cell over here gets an extra chromosome, and one cell over here is missing a chromosome. That's called a monosomy when there's a missing chromosome, and a trisomy when there's an extra chromosome. And these are called aneuploidies. Euploidy is the correct number of chromosomes. But for people, it's 46. Aneuploidy is an incorrect number of chromosomes. That is missing one, 45, and have an extra one, 47. So this can happen during either meiosis 1 or 2. Okay. So, so in meiosis 1, homologous chromosomes fail to separate. So we end up here with extra chromosomes. And we should have 2 chromosomes in each cell, right? So instead we have 1 over here and 3 over here. Then we end up with what's called N plus 1. N is a ploidy. N is a set of chromosomes. So diploid cells are called 2N because they have 2 sets. Haploid cells are called N because they have 1 set. And the ones that have an extra N was called N plus 1. So in humans, that would be 24, not 23. Or N minus 1, 1 less than N. And again, in humans would be 22 rather than 23. So it could also happen in meiosis 2. The sister chromatids fail to separate in metaphase 2. And so end up again, this causes less problems because 2 of them are the same. In the last one, all the gametes had the wrong number of chromosomes. In here, only half of them have. One has an extra chromosome, one has missing a chromosome, but the other 2 are normal. Figure 8.2.1 shows what can happen when an abnormal gamete produced by non-disjunction unites with a normal gamete during fertilization. When a normal sperm fertilizes an egg cell with an extra chromosome, the result is a zygote with a total of 2N plus 1. Because mitosis duplicates the chromosomes as the abnormality will be passed on to all embryonic cells are going to have the wrong number of chromosomes. And so if the organism survives, and many times, most times when this happens, results in a miscarriage. The embryo doesn't survive. Right? But if it does survive, some are viable, which means they will actually be born. They will have an abnormal karyotype and may have a syndrome disorders caused by the abnormal number of genes. You think, you can see how it would cause problems if you're missing a chromosome. Or maybe not, because you'd say, well, if you're missing a chromosome, it just means you have one set of those genes. What's wrong with that? Well, there is something wrong with that. And why would you having an extra chromosome? Well, it usually causes problems. There are some syndromes, and we're going to go through the syndromes of missing chromosomes and extra chromosomes. Some of them, there are no changes. The person is perfectly normal. Other ones, there are significant changes. So there's a normal sperm cell, and it shows an abnormal egg cell with an extra chromosome. And the non-disjunction happens more often in females than in males for reasons which we will talk about later on. Not that it can't happen in males, it does happen in males, but it's more often in females. So here is an egg with 3 chromosomes. In this case, the diploid number is 4. N plus 1 plus a normal sperm end up with 5 chromosomes. We're supposed to have 4. That's 2N plus 1. 2N is 4 for this species plus 1. Okay, in the case we call trisomy 21, there are three number 21 chromosomes making 47 chromosomes altogether. As I said, having an extra chromosome is called a trisomy. And trisomies of, so we have 23 pairs of chromosomes. Trisomy 1 through 17, fatal, miscarriage. These people will not be born. But trisomy 18 through 23 are viable, and some with some problems. And why that is because I think, remember, the higher numbered chromosomes are smaller. There's fewer numbers of genes, so it causes fewer numbers of problems. You know, chromosome 1, 2, 3, 4, 5, these are all gigantic chromosomes compared to the ones with the higher numbers, that I guess there are so many screw-ups because there's so many more genes on those chromosomes. But that's a speculation on my part. Trisomy 21 is actually the most common, I'm not sure why, most common of the aneuploidies. And it's called Down syndrome. It's not Down syndrome. I think that's a typo. Down's syndrome. Affects 1 out of every 700 children. It's the most common chromosome number abnormality. And it's the most common serious birth defect in the United States. So here's a normal karyotype of a male. Remember, here's a karyotype of a person with trisomy 21. There are 3 21 chromosomes. See how tiny they are. And this happens to be a female. And I think everybody knows people with Down syndrome. They're mentally challenged. You know, they have short stature. They usually, their lifespan is usually less because they usually are susceptible to early stage Alzheimer's, but they're very beautiful people. They're very nice people, and they can be sort of high function. A lot of them can hold jobs, simple jobs. And and I think they live wholesome lives, you know, happy life. They can live a wholesome, happy life. Okay. Characteristic facial features, as I said, lifespan shorter, developmental delays, mentally challenged. And some individuals can live to middle age or beyond. They're socially adept. They can function well within society. They do simple jobs. And they're actually very nice people. Okay, although we don't, well, I'm going to have some speculation on this. Although we don't know why, the risk of Down syndrome increases with the age of the mother. The fetuses of pregnant women aged 35 and older are therefore candidates for chromosomal prenatal screenings. As a matter of fact, if a woman is 35 or older and she's pregnant, she goes to her obstetrician, he is going to recommend what's called amniocentesis or chorionic villus sampling. You go into the uterus and you get some fetal cells, and you're gonna squash and spread them out, look at the chromosomes, and count the number of chromosomes. Because it's really strange that like I said, if you look at the graph for the probability of having a trisomy 21 in a younger woman, you know, from puberty on up to, you know, 20s and early 30s is very low. But at 35, it just goes like exponential. And here's here's my speculation. It's because of so we're going to get into the differences between male and female meiosis. Okay, in meiosis, in males happens at puberty. Adolescent boys don't make sperm. They're not doing meiosis at puberty. But in females, meiosis begins when they're fetuses. meiosis has already started when they're born. Well, it's arrested. It started and it gets arrested in prophase 1. So a little baby girl has got ovaries with oogonia that have already started meiosis, but they're arrested in prophase 1. And then 12 years later, under the influence of sex hormones, some of these oogonia are selected in some mysterious process to become what's called primary follicles and secondary follicles and Graafian follicles and make a functional egg, oocyte. So here's the thing. So at age 35, these chromosomes have been glued together, so to speak, for 35 years. And now they're told, now it's time to separate. Well, the longer I think the longer they spend together, I guess the stronger that glue gets, and the more likely that they won't separate when they're supposed to separate. Right? So like I said, gynecologist, obstetrician would guarantee that he or she would suggest to the woman to get tested. And if you and there's still options to abort if you want to, or at least you know that a special needs child is coming. At least you know something. Okay, non-disjunction meiosis can also lead to abnormal numbers of sex chromosomes. Oh, wait a second. They didn't talk about monosomies. So they talked about trisomy 21. Like I said, trisomy 18 through 23. Well, trisomy 18 is pretty much a death sentence. I mean, they may survive for a few days, maybe a few weeks, but they're gonna die in infancy. And as far as the monosomies, they're all lethal. Every single one of them, except one that deals with the sex chromosomes. It's called Turner syndrome. And it's only like one X, not two X's, but one. That is the only viable monosome. Okay. Unusual numbers of sex chromosomes seem to upset the genetic balance less than unusual numbers of autosomes, because some of these, there's no what's called phenotypic change. Nothing you can see from the outside. They're like perfectly normal people, and yet they have an extra chromosome, or they're missing a chromosome. So, like I said, aneuploids in the sex chromosome seem to have a less of an effect on the overall person than the autosomes. Okay, so I'm going to end it here, and we'll start off at table 8.1 in the next video, talking about the most common human sex chromosome abnormalities. Okay, so like I said, this is 106 cellrepro3, and it's going to be next one will be 4. Okay, let me stop. I'll catch you in the next video.